All right, we are ready to start. So welcome to the second day, first session of the second day of Bioconductor Conference. So we have our virtual speaker, Jacques Serizay, and he will be talking about Hi C analysis using Bioconductor. So Jacques, the stage is yours, please.
Hi, and next, thanks for the introduction, and thanks for letting me present the work I've been doing over the past few months and years, actually. So this talk is going to be, it's the first time I attend the Bioconductor conference. So hopefully you guys will be pleased with the presentation and the demonstration I'm giving. And I'll be talking about orchestrating Hi C analysis with Bioconductor. Okay, so before I get started, I wanted to point out some resources. There's a book, full-featured book that I have written, and it's available at this website. There's also a package demo website, which is also at the same on the same repository .bioc 2023, and today we're going to talk about the HiCExperiment and the HiContacts packages, and you can find their repositories again at the same link here. Okay, and there's also, I wanted to mention, there's also a Docker container that is available for any of these of the analysis, either the general OHSCA Docker image, or the one specific to the package demonstration for today demonstration. And I should be able to see questions live. So if you guys have any question, I'd be happy to answer them as I speak. So don't feel like you can't interrupt. Yeah. Okay, so what is Hi C? Hi C is, so I'm going to go through this outline, starting with chromosome conformation capture, technical aspects. It should be actually experimental aspects and technical aspects. Briefly, Hi C is a genome-wide variant of chromosome conformation capture experimental approaches. And so I'll go through what chromosome conformation capture experiments are. For Hi C, the way it works is by taking nuclei. Someone can cross-link the DNA to intact nuclei, and this leads to the formation of covalent bonds between, let's say, a blue genomic locus and an orange genomic locus in this case. That are brought in contact thanks to a protein complex here in the middle. And after a few enzymatic treatments, starting with a restriction enzyme, following with end repair and addition of biotin, eventually followed by religation, one can form these chimeric pairs of a blue genomic locus fused to the orange genomic locus. And in the case of Hi C, one would ideally want to pull down fragments, chimeric DNA fragments that are labeled with biotin, and extract those and sequence them in a high throughput sequencer. And so that's the experimental workflow for chromatin conformation capture experiments, and specifically for Hi C. And there's some variants, including micro-C and DNA-C, and less genome-wide focused approaches such as 3C and 4C. But I'm going to talk today more specifically about Hi C and other genome-wide approaches. So genome-wide, when you get the result of your sequencing, you end up with these chimeric DNA fragments being sequenced on both ends, and paired end sequencing is a requirement for Hi C. And the reason why you need paired end is because then you end up with a number of pairs here. There are 8 of them depicted here, and for each one, you're going to map the end to a genome reference. And this, in this case, pair 1 will have one fragment mapping on this position on chromosome 1 and on this position of chromosome 1. But you can have interchromosomal DNA chimeric fragments, such as pair 5 here, mapping on chromosome 1 on one end and chromosome 2 on the other end. Okay, so that is a sort of that can mapping, that mapped data can be contained in a SAM file, but typically what people like to do is summarize the result of the Hi C experiment in a pairs file, which is an example of it is shown here. It has it's a strict format that is defined by the 4DN consortium, starting with one column called pair that could be called pair ID, indicates which pair you're referring to, and then 3 next columns indicate where the first end of the DNA fragment is mapping, and the last columns indicate where the second read of the pair is located of the genome of reference. Okay, so that's a pairs file, and that's eventually when people perform Hi C, that's the sort of end point, one of the endpoints. Another endpoint is to, because this pairs file is huge, people tend to compress the data in a binarized, in a binned way. And so to do that, first one can partition the genome into fixed width bins. In this case, there's 7 bins distributed along the genome of reference that is made of 2 chromosomes. And then for each of the pairs shown here, for the 8 pairs shown here, one can then do the binning, the association of each end with the bin it corresponds to in the genome. And there's a question in the chat: when you do mapping, do you suggest people to trim their reads by enzyme digest site? If yes, do you have any suggestions for tools? So I don't necessarily trim the ends for the enzyme digestion site. One thing that is being done is when you have these chimeric reads here, what happens is that sometimes you have multiple restriction sites that are present in the same. So you have multiple religation events, and you end up sequencing multiple religation events. And that can be done. This problem can be tackled using iterative mapping. So typically, what is being done, especially on large genomes and long read sequencing, is to, well, longish, as in 100 base pair and more sequencing. What can be done is first trying to map the first 20 base pairs. Then for the reads that don't map, extend to 40 base pair and then 60 base pair, etc. And I'll mention some tools in a minute when I talk about workflows. Okay, and so this temporary representation of the pairs can be further summarized by simply counting how many times bin 1 and bin 2 are represented here. So in this case, bin 1 and bin 2 are represented 3 times by pair 1, 2, and 3, etc. And so if you summarize the data, you end up with this matrix here, which shows bin 1, bin 2, and the number of times these 2 bins are interacting. And that's essentially what people can describe as a sparse matrix format. And the description of each bin, so bin 1, 2, 3, etc. here, the nature of these bins is defined in what people refer to as the regions file, which is essentially a bed file indicating for each bin the position along the genome. All right. So when you see these two sets of files together, when you have them both in hands, what you can visualize, the way you can visualize them is also in a 2-dimensional matrix. Here, only showing the upper triangular matrix values, and so this is the direct representation in a matrix format of these data sets. And if you expand the sequencing, and you not only sequence 8 pairs as shown here, but hundreds of millions and sometimes billions of chimeric DNA fragments, you end up with a really big matrix with, of course, lots of values ranging from 0 to anything to hundreds and thousands and tens of thousands in each cell. And a typical way to represent this is through the use of a heat map. And in this case, this is the representation of this type of data, but this type of matrix, but with a lot more points. And the color code now indicates the contact frequency. And the contact frequency is scaled in log 10. This is why you have a scale with negative values. Okay, and so in this representation as a heat map, you have two different axes, but actually the two axes are identical, and this is why you end up with a symmetrical matrix. And any feature that stands out of the matrix here represent interaction between distant loci. So in this case, you have a red dot here that represents interactions between this part of the chromosome in blue and this part of the brown chromosome. All right. And of course, there are formats that exist to store this type of data. I've already mentioned the pairs file, which is shown in here. So this is a very wordy format. It's not very size efficient. That being said, you keep most of the information that you have straight out of the Hi C experiment. It is human readable, but it's not easily accessible in Bioconductor, and particularly you don't have there are no existing methods today to perform random access to these pairs files. HiC-Pro files are generated using the HiC-Pro workflow, and they're essentially these regions and counts.txt files, so they're human readable as well. Essentially, they're essentially these two set of files, and they have to go hand in hand. If you only have one of the files and you don't have information regarding the other file, and you don't know what genomic features are represented by bin 1, 2, and 3, etc. So HiC-Pro files, you do need these two files always together. Also, they can also take significant space, and this type of data only contains binned matrices. So at a given resolution, and only a single resolution, and it only contains a single type of matrix. So you can only have one single column here in the bin counts, and no type of normalization associated with these counts. All right, so few years ago, two different consortia decided to come up with their own format for more efficient storing of Hi-C data, and particularly of binned Hi-C matrices, and that led to the birth of the cool format and a Hi-C format, which are two different standards now for Hi-C binned matrices. Both are binarized file formats. One is based on HDF5, and one is a particular proprietary format that is documented, but not super clearly. They both support random access. The cool files through the RHDF5 lib, and the Hi-C files through the straw-R package in R, which is provided by the authors of the Hi-C format. And they both support multi-resolution matrices, and they can store normalization vectors, sometimes once, sometimes more, and they both have supporting libraries and APIs in Python and in the Bash terminal to interact and parse these cool and Hi-C files. Okay, that being said, so we do have these three independent formats for Hi-C binned matrices. And although there are a number of packages in R, there's no way to study Hi-C. There's no unified way of parsing these HiC-Pro or .HiC or .cool files in R. And there's even less of a unified method to parse these files. Okay, There's not either a dedicated Hi-C data structure that exists to contain data that is imported from disk stored .hic and .cool and HiC-Pro files that exist yet, and that's the purpose of this package demonstration. And there's no basic arithmetics methodology for Hi-C matrices yet implemented in Bioconductor. So that's what I wanted to talk about, and I'm going to introduce the OHCA for Orchestrating Hi C analysis ecosystem in R and Bioconductor. Okay, starting with the HiCExperiment class of object. This is an S4 class that essentially is based on genomic interactions and the BiocFile class. It works by importing data from contact files, and these contact files are another class. They can be cool file, HiC file, HiC-Pro files, or pair files. And all of these files act as, these file classes act as pointers to disk-stored HiC data files. And the HiCExperiment package defines an import methods to parse these contact files into a HiCExperiment object. And importantly, this import method supports random access for HiC and .cool files using a unified syntax. Okay. Now, if you're interested in studying HiC, but you don't have yet your .hic or .cool files or pairs files, and you want to actually process from scratch the FASTQ files, there's a solution for this now all embedded in R. Using HiCool, one can leverage the HiC stuff, which is a lightweight Python HiC processing library that is going to take your FASTQ files and pass them into pairs and further in .hic and .cool files. And this is this package is based on Basilisk, and it wraps, so it creates a self-managed Conda environment to install the HiC stuff package from Python. So the different processing steps undertaken by the HiCool workflow are the following. You take FASTQ files and a genome of reference. It can be either a local FASTA file or a genomic ID. And each FASTQ file R1 and R2 is going to be mapped to the genome reference independently, and then the pairs file is going to be reconstituted. And so at this step, to go back to the question from the chat, the trimming of eventual trimming of the reads is done internally under the hood, so you don't have to take care of the different options for the mapping of your data, but you do have a hand at some of the controls for this processing still. Okay, and once you have these pairs files, the HiCool workflow sorts these pairs and then filters them using specific criteria, and then eventually parse them into a cool file and a multi-resolution mcool file and .hic files. And at the same time, it generates an HTML report. And all of this is triggered really with a single function. It's the HiCool function. So it really is a single function that triggers all of this workflow. And we'll see how it goes in the demonstration. And how is it normalizing? The normalization of this data is done through matrix balancing, specifically using the algorithm published in Cornac et al. in 2012. Actually, probably I should have added it. Cornac et al. I think it's PNAS. I'm not sure about the reference. PNAS or plus 2012. Okay. But essentially, it's the normalization, one of the generic approaches for matrix balancing that is implemented in R. similar to the ICE. Okay. And so once you have these .hic or .cool files, you can, as I explained, import them in R using the HiCExperiment package. And once you have these object HiCExperiments, the package HiContacts is here to provide you with some analytical tools. First, you can do some genomic arithmetics for HiC maps. You can also look at the analytical tools specifically dedicated to genomic interactions. There's some implementation of algorithms to annotate 3D features, including compartments, topologically associating domains, and chromatin loops. Also there's extensive ggplot2-based visualization methods shipped in this package. Okay. And two companion packages for DNA data and DNA zoo data have been put together. They're exclusively data packages. They don't provide any type of analysis functions. They only serve the purpose of opening a gateway to data provided by two major HiC consortia, so 4DN and DNA zoo. They're experiment hub-based file accession, and they support BiocFile cache. So that means if you're interested in one of the data sets published by or generated by one of these two consortia, then you can use these packages to access the data directly in R, and this will fetch the matrix, the HiC matrix files from their server down to your local computer, cache this file, and then automatically create a pointer for a contact file for these cached HiC matrices. Okay, and all in all, you end up with these 4 different packages interacting with each other that help you go from FASTQ files to endpoint analysis, and we'll see in the demonstration how this goes from beginning to the end. Okay, and I guess I can start with description of the structure of the HiCExperiment object. Briefly, the HiCExperiment object is created upon importing of a contact file or pair file in R, and you can specify a focus using a UCSC style genomic coordinates. If you don't provide any, imports the entire HiC matrix in memory, and you can also provide a resolution if your contact file is multi-resolution based. So namely mcool files and .hic. Okay, and this creates the HiCExperiment object, which contains a number of slots. The first one are file name, which is a string character pointing to the location of a disk stored file. So it's not necessarily a cool file, it can be a HiC file or HiC-Pro files, etc. You can also have optionally pairs file associated with your contact matrix file. And if you have provided a focus, you can access this focus slot in the HiC kept in the HiCExperiment object. This HiCExperiment object also contains the available resolutions. If there are several of them, then you'll see them listed in this resolutions slot. And you'll also have a resolution singular form indicating at which resolution the data actually has been imported in memory. And the data itself is stored as genomic interactions, which you can access through the interactions function getter. And so you have interactions starting with the first end of the interaction and the second end of the interaction. And you have companion scores, which you can extract on their own using the scores getter. And these scores can be numerical matrices, numerical vectors. So they would be raw counts, for instance, in most cases, normalized counts as well. But you can also store any type of other numerical values such as p-values or fold change if you're interested in fold change and comparing two matrices. You can also store categorical values if you want to differentiate interactions based on categories. The scores slot really can contain whatever type of vectors you want. And finally, this is a accessory slot called topological features that can be used to store and synchronize topological features connected with your data, such as chromatin loops, or, domain borders, or compartments, really whatever. This is a list, and you can put whatever genomic ranges or genomic interactions you're interested in. And of course, you have also some metadata associated with this HiCExperiment object. Okay, and because this is heavily based on genomic interactions, which is itself based off of genomic ranges, class of objects, I tried to recapitulate the different methods that are available for these genomic interactions and genomic ranges. So you can use the square brackets to subset a HiCExperiment imported in memory. You can use the dollar to access the different columns associated with the different regions of the HiCExperiment. You can access the seq info to get the information on the genome, the chromosome information and length, etc. And I wanted to skip this. So maybe now I'll move on to the first vignette of the workshop, and it's available here. I'm going to move over to our studio, and hopefully everything works. So the parsing Hi-C data vignette is here. Again, you can just click on the link from the presentation, and it will point you to this document in a website-based format. Just before I get started with the demonstration, everything here is done through a Docker container that I've loaded and got it started using this command here. And the Docker container image is located here. And there are currently two different versions, one for Bioconductor release 3.17 and one for the development version of Bioconductor, which is updated in a daily fashion. So if you want to replicate this demonstration, or if you want to get started quickly with the Hi-C analysis, feel free to fetch this container image and get started. Okay, so I'll first start with HiCool. I can load the HiCool library, and I'll skip over this. The HiCool library provides me with the HiCool function, which does the help menu, indicates the different, it's BMC Genomics from Cornack et al. It indicates the different steps that are being performed under the hood, and the different arguments that one needs to provide is at least a read1, read2, and a genome reference. And restriction enzymes can be specified here, and there's a number of extra options that are documented in the HiCool arguments help here. Okay, so in my case, I'm going to fetch R1, FASTQ R1 and FASTQ R2 files from support data package called HiContacts data. And HiContacts data allows one to fetch toy example data sets. So R1 and R2 are now two files that are cached in R in my local computer. I can check actually maybe the read lines of R1, and I'll do n equals 10. Yeah, and we can see that line 1, 2, 3, 4, etc. corresponds to the first entry in this FASTQ file, and it works with zipped FASTQ files as well. So you can copy and paste here the HiCool function using R1 and R2 cached files. Restriction enzymes that have been used are these two ones, and the genome reference is the genome reference for yeast. So in this case, you can put only R64-1-1, and automatically the bowtie 2 genome index will be recovered from the iGenome Amazon bucket. So it gives indication about the different steps being performed, and in a second it'll be finished. And it tells you that it's generating a HiCool report that may take a while, and once this report is generated, it prints it here for the path of the report. And now I have a HiCool file, which is a class of cool file object, pointing me to the location of the mcool file that have been generated in this workflow. And if I go to my files, I go to HiCool matrix, and now I have so the new one I generated has this hash code GV something. So it's this one. It's this guy here. Which I can't open in plain text, but it's available here. Okay. So now, if I want to import this mcool file, one way to do that is to use the HiCExperiment function from the the import function provided with by HiCExperiment package. And now, if I do import of, I can copy and paste this this path here, and I'll create a variable called x. Okay, and we can see that this has loaded the data, and if I print x, this is the output summary of a HiCExperiment object. Now, what I did here is to import the mcool file directly. I can actually use the cool file class of object to import it in a more robust way. It has more checkups associated when you import this HCF object, but it does the exact same. The exact same operation is performed. Okay, I can also check the available chromosomes for the HCF object, and I can also check the available resolutions for the HCF. And that can be done before loading any data from this mcool file in memory in R. So it's an easy way to know the length of your chromosomes before passing the entire or any part of the data in R. Okay. So I said you can import using the import function. And this imports the entire chromosome. So you can see in the focus field, which you can access using the focus. Whoops. I'll create a HiC. If you check the focus of the HiC object, it's null, meaning the whole genome has been imported in memory. If you check the resolutions available, you can see that there are that many, so 5 different resolutions available. And by checking the resolution of the HiC, we get that it's imported at the 1 KB resolution. Okay, and all of this is actually quite visible when you print the summary of the HiC data. So now the actual data contained in the HiCExperiment object is located here. Interaction and scores, and one can simply access them using the getter function scores. And if you want to get more specifically the count values, you can say scores count, and here I'll say 1 to 10, and I see that I have 10 first values of my scores. Now I can also do interactions of this object, and this returns a full-featured genomic interactions, which has, in this case, 56,000 interactions listed. And under the hood, it also appends the scores to the actual interactions stored in this Hi C object. So if I actually get the actual interactions slot, you can see in of the Hi C object, you can see that it doesn't contain the scores, count and balanced. But as a convenience, the getter function interactions just appends the two extra columns, count and balanced, which are the two scores listed in this object. Okay. Of course, one can also import, let's say you're only interested in chromosome 1. You could say, I want only the chromosome 1. And in this case, you don't end up with 1,200 regions anymore. You end up with only 231 regions, and the focus now is chromosome 1. And actually, if I were to check the interactions, now I have a lot less interactions, only 605 individual interactions. Okay, there are alternative ways of importing the function. One can specify a full chromosome or only a part of a chromosome. If I want the 101st kbs, I can do this. If I want interaction between two chromosomes, that's also doable. Interactions between chromosome 1, first 100 kbs, and chromosome 2, 200 first kbs. This can be done using this this approach. And Jessica Holmes says, I can't see. Can you see it back? Can you see the screen back now after I switched? Oh, so it's only Jessica who's having issues. Maybe try and reload the web page. Ah, awesome, great. Okay, so that's so this import. Also, what I haven't talked about is the resolution argument. If you want to change the resolution and import the data directly at resolution of maybe 2 KB, you can do that. And rather than having 300 regions, you have 150 because the resolution is twice as big as originally imported. So by default, the resolution that is going to be imported is the smallest available resolution. So in this case, it was 1 KB. Okay. So that's how you you navigate with the data. Once you have imported this data in memory, what you can, and I'll do it now, X, okay. What you can do is actually subset this data using subset by overlap, using the same syntax than what is typically being done for genomic ranges and objects. Oh, I need the genomic ranges package. Okay, so I can subset by overlap for such genomic ranges object, and there are further subsettings once the data has been imported in memory. There are further additional subsetting methods that are documented in the online book. Okay, so that's for parsing Hi C data. Is this subset by both ankles? Actually, yeah. So to go further, you can say you can specify the type, and you can say either actually maybe the easiest would be to show you this. Yeah. Here, so you can specify whether you want subsetting by overlaps with type within. In this case, it will subset using the two ankles. If you subset with type equals any, it will subset using any overlap between the ankles of the interactions. Okay. So moving on to, I don't know until when I have time to go, but do interrupt if I'm running over the next session. So to give examples of how to manipulate Hi C data once it's imported in R, you can navigate to this. Oh, first a visualization of the data can be done using HiContacts. So this is the second vignette here. I'm going to import this and create a Hi C object coming from a yeast data set. So I now have my Hi C object, which has imported a 2 KB resolution over chromosome 5. And if I, plain and simple, I want to see this matrix, I can use the library HiContacts and the provided plot matrix function. And this will pop up a Hi C matrix with chromosome coordinates, the key indicating the color scale and the boundaries of the color scale. What one can use is arguments to tweak this color scale. If I want to color from -5 to -2, for instance, this will change the color scale accordingly. Of course, in this case, the boundaries are not ideal. One can also change the color map by providing a number of vectors to indicate a gradient. In this case, this function here provided by the HiContact is just colors ranging from blue to red, going through white. And final argument important for plot matrix is the scale. In this case, automatically the scale is switched to log 10. So by default, this argument scale is in log 10. But one can also plot a linear scale. I'll erase this. And this scale, in this case, the scores are of course going to be from 0 to positive values. Because the counts, either raw or normalized, are always positive values. Which leads me to the last important argument of the plot matrix function, which is the use scores argument, which can be used to specify which type of scores one would want to print. So again, scores are listed in Hi C. And in this case, it's count and balanced. And by default, it plots the balanced counts, but you can change this behavior using the use score argument. Okay. So there's a question in the chat asking, could I plot the data for several chromosomes in one page? So I'm not sure that this answered the question, but in this case, I'm importing the full Hi C data set from this cool file object, which is cached in my computer at resolution 4 KB. And then what I did then was to subset the full Hi C object, which is over 3,000 regions, to a Hi C subset object, which is now only 463 regions, being chromosome 3 and chromosome 4. And that was done using the square matrix, the square bracket notation here to subset. And so this Hi C subset then can be plotted as here, and here you can see interaction between chromosome 3, chromosome 4, and between chromosome 3 and chromosome 4. Now, if you want to only see the interaction between only chromosome 3 and chromosome 4, so this interaction, one would need to import the data set using import Hi C focus is 3, 4. Oops. Oh, it's CF. And now we can see that the interactions are only between chromosome 3 and 4, and there's that many number of regions corresponding to all chromosome 3 and all chromosome 4. Okay. So moving on with the visualization, going to investigating. A number of arithmetic operations are available. One can normalize on the fly imported data using the normalized method. I'll go over through the last slides, and then you guys can go through the vignettes at your own pace. In so arithmetic operations available are to divide different two different HiCExperiment objects, aggregate all the snippets, merge HiC experiments, do autocorrelation, and computing observed over expected contact matrices. Everything is documented in this vignette and in the online book. One can also compute the distance law or the distance-dependent interaction frequency using this getPS function, virtual 4C profiles to show the interaction profile of a given genomic locus with the rest of the genome, and the ratio of intrachromosomal versus interchromosomal interactions, and finally the distance-dependent interaction frequency for sliding windows along the genomes. All these functions are available to look at genomic interactions. And finally, one can look at the different topological features and annotate them using get compartments, get diamond insulation, and get loops functions from the HiContacts package. And with that, maybe I'll finish here. There's a number of coercion methods, but I'll skip this interoperability section, and this is all documented in the online book. And I'll wrap up. And with the could you show a screenshot for the virtual 4C plots look like essentially. Well, it would be this. I think I should have it somewhere real quick. In here, yeah. So this is virtual 4C plots computed for the region, this genomic locus over the entire genome, and you end up with a genomic ranges object. So you're then free to perform plotting as you may wish using ggplot. And that's the interaction of the chromosome 2 with chromosome 1 segment, chromosome 2, 3, 4, etc. Okay, so yeah, just going back to acknowledgement slides, and I'll be all for me for this demonstration. So huge thanks to of course all the bioconductor packages and infrastructure, more specifically the interaction set which defines the genomic interactions data structure, and the BioIO package for the import generic method. And thank you all of you guys for your questions and interaction and attention. Thanks, guys.
Right.
Everything is available online as well.
Terrific presentation. So we don't have much time, but if you guys have any questions, you can reach Jacques. He's one of the participants, so reach him by platform, ask any questions, and definitely follow up on his material. So on this, let's close the session and transition to the next one. Thank you.
Thanks. And Asa is asking, is there a function method to also detect, visualize sub? So Asa, the sub compartments, they're not really tackled in this. Sorry.